Hello, am I audible to
Hello.
you? Hi, hi.
Hi, I'm
How are you?
Wu Zhongtong.
Am I audible? Hello.
Another Yuan. Hi.
Hi, hi, Chiung. Am I audible? You can hear my voice?
Okay. Yes.
So should I start how what you guys have to do?
Okay.
You guys are getting me?
Okay. Okay. I'm ready.
Yeah. So if you don't understand anything, just do stop me and ask me. But it's a bit difficult for me also. These currently I'm also learning these things.
Uh,
So what we will do in this, like on data mining is like we will do all these things on all that analysis using bioinformatic tools. You know what is bioinformatic tools? Like online, all the online tools that we can run any analysis using codes or help of AI. Okay,
oh.
so here
Okay.
we'll do that. We'll use that some like or it's will not use any code or any AI system. It's already websites are made by some scientists on the internet. So what we will do, we will take out the data. We have some values, some relation values, some p values. So we will put those values on the tools, and that website or any software will give us the information regarding that. So first what we will do this week is you guys have choose your cancer. So I will give you some data, like you have to go to a website. I'll send you the website link. I'll also show you now, and then you have to take out the data list. There will be some thousands or 2,000s or 5,000s of genes. So what you have to do, you have to first see like which genes have zero publication on internet, like PubMed or Google Scholar. So if no one is working with this gene, so after that you should start working on that gene and do some publications, because if no one have used that gene, it's a like a pure gene. So you can the chances of publications, your paper publication will be high, because this gene was just first time. Okay, so you guys have to do this. So now I will start how you will go to the website and you will download the data. So till here, do you have any query, any questions?
No. No.
Okay, so let me share my screen. And yeah, so you can see, huh?
Yes. Yeah.
Yeah. So the first website is called GPA. I will share you the link of this website, GPA. So as already told you that you have to find out your genes. So on this is the website of GPA. What you have to do, you have to go on generals. No, differential. Yeah. So you have to come to differentiate genes. Okay. So we also call them DEGs, differential express analysis and differential express genes. So these DEGs are like, you can say that they can like help in getting overcome from cancer, or also use in cancer, like it will cause cancer in your body as well. So uh, you have choose breast gene. So like you will come here. If you don't know the shortcut of your cancer name, So you will come here and you will see that BRCA is the shortcut of breast invasive carcinoma. So
Okay.
you have to put the shortcut over here. Okay, just note down this if you have pen or paper, and you have to choose this Lima method, differential method. You have to choose Lima, and then you have to choose both overexpress and underexpress. It means that the gene that you are going to use in breast cancer is overexpressed in cancer stages or underexpressed. Like we have four type of cancers, four stages: stage 1, stage 2, stage 3, and stage 4. So at a certain stage, the gene is overexpressed or underexpressed. Like if it's underexpressed, it is non-cancerous. And it is overexpressed, then it is cancerous. It can cause cancer in your body. So you have to use both the genes, okay? And then you have choose the code of your cancer, like breast cancer, Lima method, and then you will put take out the list. This is the list of total genes that can cause like or that are involved in breast cancer. Okay, these are the name of genes. Gene symbol is the name of gene ID, the tumor medium, the normal tumor, log2 FC value. You can just check it what is log2 FC value, agent P. These are the numbers that will help you on the bioinformatics tools to get analysis of the genes. Okay, so you will get 3,599 genes. Okay,
Okay.
so now what you have to do, just first of all download this table, okay, and then just let me just. So you have to import. It's depend on your computer. Either the data downloading in text format or it's downloading in your this Excel format. Okay. So if it's not in Excel format, just take it in Excel. Uh, you have to just import it in Excel. Uh, download tip chain. So here, like after downloading the data, so we have all the genes. Gene symbol is gene name. You can say that this is the name of the genes that are involved in breast cancer. Okay, gene ID, tumor, normal, log2 FC. So what you have to do is now you have to adjust p value. This is also called p value, ADGP. So it's a p value. What you have to do, you have to go to a number filter, and you have to do is less than or equal to. You have to make it less than 0.05. 0.05 is like we will take a p-value till 0.05. After that, like we used to say, there's a gene called down regulated and up regulated. Down regulated is also like it's gene who have low less separate less like I told you that overexpressed and less expressed. So down regulated are less expressed about like those gene who are down regulated, so they don't have too much expression in breast cancer. They don't show too much express or they don't involved in too much in breast cancer. But those who are upregulated, like less than 0.05, is they are highly regulated, like highly expressive in breast cancer. And we have to find highly expressed genes that are not used till date in any publication for online analysis. So we will do it less than and equal to 0.05, and we'll do this. So you will get the value less than 0.05 in this, okay, here 0.05. So now what you guys have to do is you have to check all these gene on PubMed, whether they have publication or they don't have publication. Okay. Before that, before publication, you have to focus on one more thing, is that create a column and make a column says cancerous slash non-cancerous. Okay, and another which say significant and non-significant. So cancerous, non-cancerous, you guys know what this means. Cancerous is like the gene is cancerous or non-cancerous. Whether this gene can cause cancer or cannot cause cancer. Significant and non-significant means that this gene is significant, like it's cancerous, and it's also significant, then this gene is highly chanceable of getting breast cancer in human. So if the gene is cancerous and significant and zero publication, that gene we have to use for our analysis. You have to just choose few, one or two genes out of this 3000 something genes. You have just take out one gene. So what you have to do, this week I'll give you this work for the whole week, or whenever you will finish, then after the next meeting we will review this. So you have to do till here, you have to do this. You have to make two columns, cancerous and significant. So now I will tell you how to find out whether this gene is cancerous and significant. So any doubts till here, guys, any doubts?
Okay.
You're getting my English also. Should I speak some slow, or it's okay?
Yes.
Yeah. So now how we will find cancerous and significant? Okay. So just copy this gene and go to the website GPR. So now you have to go to expression DIY. Here expression DIY, you have to go to box plot. Just note it down, okay? So expression, then box plot. Then over here, here you have to put that gene name. Okay, this gene name. So when you put here the gene name, then you have to choose your cancer. Like if you have other cancer, you have to choose, like we are using breast cancer. So you have to choose breast cancer and add.
Breast cancer.
Here it will appear. You just leave this as it is. You don't have to touch anything. Just put the gene name, choose the cancer, add and plot. So when you will plot here, it will come.
Oh,
So, how you will understand that gene is cancerous and non-cancerous? So, I will tell you, this is a box plot graph. We will use this also for our review paper. So, the red one, this is cancerous gene,
breast
and the
cancer.
black one is normal gene. Okay. So
Okay.
just view this line, the line in between black, the normal cancer. This is called tumor number, tumor and number is normal. Normal cell tissues and like normal cells or normal, you can say genes or tumorous genes. These are the expression of genes. So they are 291 normal and 1085 cancerous. But this gene is significant and non-cancerous. How? Because see, the cancerous, you can say the cancerous gene and the normal gene is higher than cancerous. This value, the non-cancerous value is higher than non-cancerous value. Sorry. So
Oh,
look, this is up and this is down. So this gene is non-cancerous. You cannot use this gene. And how you will find the significant and non-significant? So you will see, like if you go for other gene, so this star, you can see the star. So this
okay.
star will tell you the gene is significant or not. So if there is a star like here, in every box plot there will be a star, or no, there will not a star. So if there is a star, it means that it's significant, and if there is no star, that means it's non-significant. If there is a gene, assume if there is a gene whose this red one is up and the gray one is down, so it's cancerous. And but that this star will not there, so we cannot use that gene because it's not significant and it will may fluctuate further in our analysis. So if our gene looks like this, look, the gene I'm putting is btg3, it's my
G3.
gene, and I'm currently doing on gbm, so I'm working on gbm. So I will add my gene and I will plot this. So see, mine is cancerous. The red one is
Oh,
upper than the gray one. It's cancerous, and look, it's significant also because it present star. So we have to find out this kind of gene this week, like if it's cancerous or significant or non-significant. This kind of genes will work for our research, further research. Any questions, guys?
no.
So
So far, so
no.
good.
So now I will put a gene and you guys have to tell whether it is significant and non-cancerous. Okay, guys have to answer. So I just plot it. Now tell me, it's cancerous or non-cancerous?
Non-cancerous.
Yeah, and significant or non-significant?
It's not so significant.
Non-significant. Correct, correct.
Okay.
So we don't have to go with these genes because these genes are non-cancerous. Like they are cancerous. They are cancer gene, but not that much problematic. It will not make any problem in your body. Like in future, they will get converted in cancerous cells and get a cancer. No, they are very like low cancerous probability to get cancer in your body. So this week we have to do things like this. Just a second, where is can, yeah. So like this one was non-cancerous but significant.
Significant.
Okay, so we have to do this all in this. Don't forget to set p value first. Short largest to smaller, then go and filter it less or equal to 0.05 and okay. So you have to do this work this week. Give me some significant and non-significant. When you will complete this, then what you have to do is, I will suggest you not to go for like 3000 genes. What you will do, fill this full till 200 will be okay. Not will not go for like too much. First we will go for 200, like if you can done in 1 or 2 days, like 200. If we will get our gene in top 200, like the gene will be cancerous and significant and also zero publication, so we will use that gene for further research on top 200. If we'll not get on 200, then we will go further more. So first you guys have to do top 200. Now I will tell you how you guys will find out that this gene is like good for your future references, like into top 200 if you get any genes. So like we will take a gene called KLK8. Okay, suppose this one, this gene is cancerous also and significant also. Okay, now we have to just like in top 200 we have to only search for those genes who are significant and cancerous for research. Okay, so you got like 10 cancerous and significant out of 200. So what you have to do is you have to copy KLK8. Okay, now uh, family member, yeah. Before that, before searching that, wait, let me remember that. Should I we do that first? Format. Yeah, it's a short trick. Like it's my trick. People used to do first. They will find out the family, but my trick is like directly go to PubMed. Okay, PubMed. Just search KLK8 and write breast cancer.
Breast
Okay, and
cancer.
enter. You will get 10 results. Look, 10 people have published paper in KLK4 breast cancer. So we cannot use this gene for research. Mark it red, like whether it is cancerous or significant, but we cannot use because already used by someone. Then we will go for some other genes. Like we have to do same with every gene which are significant and cancerous. We don't have to do for every cancer, only for those who are cancerous and significant. Then go to PubMed again. Just put out down the gene and search. Like this have 17 research
17.
papers in breast cancer. So again we cannot use this. So just like an example, like you use some, like I'm giving you example how you will get to know that this gene is, you can use this gene as a research for. So like this is my gene, and like write your gene over here, then write the breast cancer and search. Look, it says your search was proceed with automatic term mapping because it's zero results. Okay, no result found for my cancer gene in GBM. And guys, you have to write breast cancer or BRCA also in front of your gene. Okay, BRCA. So it shows zero result. So it means that yes, this is the gene that we can do research. So if this is the gene that we can do research, mark it green and go for next. If this one is also good as zero publication, mark it green. Then go on in top 200. If that is cancerous, significant, and zero publication, you guys have to mark it green and go further and check all those. If you got like five zero publications, then we will find out, uh, then we will find out the family members. Like we have to see their family members also. Like every gene have their family members. Every gene, like they have hundreds of family member, five family member, two family member. So like out of these five gene are green. Okay, so we will find out the least family member. Like if someone have three family members or four family members, then we can use that gene instead of those who have 18 or 19 with zero publication, because your result will be go so long if you have 18 family members. Okay, so this is the basic starting of your work. So do this, find out the zero publication genes online, and then we whenever you guys will finish, just text me, and we'll have another meeting on weekdays. So when you will give me this data, so I will review it, and I will tell you if there is any mistakes, and then we will go for the next procedure, that is family finding. And next time we will find out that you have this gene, specific gene for your publication or for any bioinformatic tools. Okay guys, any questions, any
So,
doubts?
so we basically check nobody have been researched on PubMed, and then
Yeah,
we mark the table as green.
yeah, because
Okay.
everyone have done so much of research on that paper. But you can, like, it's like it's your basic, you have to find the zero publication gene. But when you will get experience in further research, so what we guys are doing, like, we are doing bioinformatics. Okay, so like,
Okay.
there is assume there is a gene, and there is a two analysis of any genes. One, it's a wet lab that you will guys going to do in June, July, like next month when you will come here, and a dry lab.
Yeah.
So what we are doing is dry lab currently. It's a dry lab analysis. Dry lab means like we are doing on computer. We are not using any lab. So
Oh,
some
okay.
some genes have wet lab analysis only. Assume there is a gene, and that gene is only done dry lab, wet lab analysis. They have done research in wet lab only, no dry lab, no analysis using that AI tools or any online tools. So we can use that gene also for publication. But when you will get experience, then you will get to know how to do check that also. But currently, I prefer you guys should find zero publication genes on PubMed, and then we will go further research on that. It will not take too much time. I was using to learn many things. I will tell you basic things, so we can, if professor allow you, maybe till your August mid, we can complete all the research, and we can, like, if professor allowed to write you a research paper, You can write a research paper on dry lab also. And I'm also writing on paper right now on a dry lab things because my wet lab is still not started. Like next month, I'll also start my wet lab. So let's see. We will do further analysis on that also. Any other questions?
Nope.
No.
So you got it. It's easy.
Yeah.
Let's
I
give it
will
a
try
try.
my best.
Yeah, when you will get to used to it. It's in starting, like when I started like 4 5 months before, it was so complicated to me. But now I am used to that. So if my eyes were closed, I can do the research. So you will also get to used to it. It's new for me also. I never think about this point even in my life. If I was know before, like you guys in bachelors, and I'm just going to do my masters in September. So you guys know
Oh.
till now like how to publish a paper. You will get to know soon. I'll give you some my experience also to publish a paper. If seriously, if, I know that if by dry lab I can publish a paper, so when I completed my bachelor's 3 years back, I can write 5 6 paper until here. But I don't
Wow.
know that when research are so easy, like these things are so easy.
Okay.
So but like I'll help you to do these things. And if you have any question, you can just line me.
Okay.
I'll try my best to answer you as soon as possible because I am doing research on my things. Like I can show you. Currently, I was just finalizing my papers. I'll show you that there is another things. These things we have to do further, like downloads. I'm just currently working on two genes. This is first and second gene. I will show you GBM. So these are the analysis that we will do furthermore. Like still, it's like not too much. So we will see this also after final genes. We will see the survival. What is the survival rate of a person? Like these are the survival rate and these are the days. If a patient survival percentage is 1, so how many days he lives? I will teach you the graph also, how to read this graph. We will do this analysis also. And these all research we have to do. Like this is a relation correlations, like what my genes have correlation in, like whatever gene you will see. So their family, my gene have 4 family members. So the least family member you will choose, it's better for research. Like it's not, it's easy to read the graphs also. I'll teach you further like how to read graphs. So these are the protein interaction, the protein in your genes, how they interact with each others. Like
Oh.
my protein is interacting with other proteins, then they are like, you can see the boxes. So these are my gene family, and they are not interacting with each other. Like they're not interacting with each other with any uh, personal nodes. These lines are called nodes. Okay, they are using some other genes to contact each other. If you can see, this one is contacted with him, then this one is contacted with him, this one is
Oh
contacted
yeah.
with him, then this. So they are not contacted with each other. Like you will get to know more about this when you will do this research. These are very easy. You have to just put data online, and then like this is a human tissue. You can see it's a human, normal human tissue.
Mm.
Like how it's not visible clear because it's a photocopy, like printout from the website. When we will run this website, you will get to, when you will zoom, you can see the cell and the cancer over here. It's not a, like if you will find a cancerous gene, you will see the cancer, how tumor looks like on the 4x zoom. So we will do all the basic analysis. These are all the analysis that I have done from 4 5 months. These are, and basic things, guys. Do one thing, go to PubMed and find some research paper. I'll find some research paper and send it to you regarding some bioinformatics tools, so you can read papers. Because only if you read papers, then you will get to know more about the cancer and other things. So I will send you some basic papers, how you can like do bioinformatics tools. What are the terms that you will hear first time and you need to google it, like p value, adjacent value, log fc. What is log fc? These things, like you have to do for research now. So any doubts? Nothing?
No.
No.
So best of luck. Complete your target soon, and find out the gene as soon as possible so we can do the research.
Okay.
Okay.
If you have any query, any doubts, like you just send it to me if you forgot anything. So I will just send you line, like what the things you have to do. Just download the data. If you have any problem, just text me.
Okay.
Okay, so much appreciated.
No, no problem, no problem.
Thank you.
Okay, guys, see you.
See
See
you.
you. Oh my god.
完蛋！哎、我
哎、我们用赖的好了，我怕他等下突然进来。
好，OK OK，拜拜。
拜拜。